bubuksan ang isipan. Sa science o agham, tayo na!
Mendoza, and I am the one who assigned to discuss the timeline of science and technology in the field of biology. So before I start, I want to ask someone who has the knowledge to 1628 blood circulation. Yes, Ms. Maskariño, please share your knowledge.
Hello, Ms. Reporter. Blood circulation.
In 1628, William Harvey, a British doctor, has performed multiple dissections on dogs. He also had demonstrated his experiments before the other surgeons. Harvey has shown that the blood circulates in two loops, pulmonary and systematic. He has also discovered bulbs in veins and determined that blood can only move in specific directions in the body. Understanding how our blood vessels work was crucial for medicine and physiology. It has impacted treatments of the time and raised the interest in anatomy.
Thank you for your meaningful knowledge. So now let's proceed with the discussion process.
1665. The first description of cells. The cells was first discovered and named by Robert Hooke in 1665. He remarked that it looks strangely similar to cellula or small rooms which monks inhabited, thus deriving the name. However, what Hooke actually saw was the dead cell walls of plant cells, or the cork, as it appeared under the microscope. 1758. Classifications of life. Carolus Linnaeus is a Swedish scientist that proposed a universal system of classifications of living organisms. The new system was hierarchical, Latin-based, and allowed easy communication between scientists. Each species was given a unique binomial name and belonged to a particular genus, family, order, class, phylum, and kingdom. The existence of the classification system has made the study of life significantly easier and also allowed to study evolutionary relationships between organisms in later centuries. 1838 to 1858. The cell theory. The cell theory was produced in two steps by Slayden and Swan on 1838 to 1839, and then remarked on 1855 and Virchow on 1855 to 1858. This theory claimed that cells were universal and microscopic entities constituting living beings, and a cell was always produced by the division of another cell. 1861 to 1862. The germ theory, or the pasteurization. French chemist Louis Pasteur developed modern germ theory. He proved that food spoiled because of the contamination by invisible bacteria, not because of spontaneous generation. Pasteur stipulated that bacteria cause infection and disease.
So in 1866, the law of heredity. So who wants to further knowledge about it? Yes, Mr. Consignado, please share your knowledge.
The laws of heredity.
1866. The laws of heredity. Gregor Mendel, an Austrian monk and a mathematician, have postulated the mechanism of transferring inherited traits from parents to offspring. He has used pea plants for his experiments and has discovered the existence of dominant and recessive alleles. Mendelian genetics is only one aspect of the complex mechanism of inheritance. However, it still plays a massive role in breeding programs in agriculture and understanding human disorders. Mendel also introduced statistics, which is now widely used in all sciences.
Thank you for sharing your knowledge, Mr. Consignado. The next is
1883. The phagocytosis. Ilya Elim Mechnikov has discovered the existence of specialized cells in animals, both vertebrate and invertebrate, that were responsible for eliminating pathogens and dead cells. Then Meshchikov's discovery has started immunology, wherein it is the science devoted to the study of defense mechanism against pathogens inside the body. Immunology helps significantly in our fight against pathogens and vaccine development. 1892, the tobacco mosaic virus. Two scientists contributed to the discovery of the first virus, the tobacco mosaic virus. Ivanowski reported in 1892 that extracts from infected leaves were still infectious after filtration through a Chamberlain filter candle. Bacteria are retained by such filters. A new world was discovered, filterable pathogens. 1928, the antibiotic or the penicillin. The accidentally discovery of a moldy petri dish in 1928 kick-started a 20-year-long journey to develop the world's first mass-produced drug that could clear bacterial infection, the penicillin. 1950, the mobile genetic elements. For much of the 20th century, genes were considered to be stable entities arranged in an orderly linear pattern on chromosomes like beads on a string. The late 1940s, Barbara McClintock challenged existing concepts of what genes were capable of when she discovered that some genes could be immobile. Her studies of chromosomes breakage in males led her to discover a chromosomes breaking locus that could change its position within a chromosome. McClinton went on to discover other such immobile elements now known as transposons. She also found that depending on where they inserted into chromosomes, these mobile elements could reversibly alter the expression of other genes. She summarized her data on the first transposable elements she discovered, the AC and DS, in a 1950 PNAS classic article, The Origin and Behavior of Mutable Loci in Maize. Although their existence was accepted relatively soon after maize geneticists, the widespread nature of mobile genetic elements and the implications of McClintock's discovery took decades to be widely recognized.
The next is DNA structure. How about you, Mr. Olila?
I'm sorry, Miss Reporter. Can you repeat the question?
Do you have knowledge about DNA structure? Please share with us.
Yes, a little bit. DNA structure.
The discovery of 1953 of the double helix, a twisted ladder structure of deoxyribonucleic acid by James Watson and Francis Crick marked a milestone in the history of science and gave rise to modern molecular biology, which is largely concerned with understanding how genes control the chemical processes within cells. In short order, their discovery yielded groundbreaking insights into the genetic code and protein synthesis.
1970, E. coli. Bacterial transformations have been around since the 1920s. Escherichia coli was being utilized as a model organism in microbiology and other biological fields for most of the 20th century, but was discovered intractable to transforming until Morton Mandel and Akiko Higa were able to induce it to take up DNA with the use of calcium chloride. The discovery of artificially induced competent E. coli cells created one of the easiest and most efficient transforming bacteria, which allows for even simpler cloning methods in all biological science. The use of E. coli has only grown in popularity as one of the most common model organisms in science, and was one of the first organisms to be completely sequenced in 1997. 1983 PCR. Few discoveries have revolutionized their field as much as polymerase chain reaction or the PCR. Likewise, PCR owes its own revolution to the previously discovered thermally stable DNA polymerase. Prior to carimolysis, work in inventing an enzymatic assay to utilize a DNA template, primers, and heat cycles, first described in the Journal of Molecular Biology in 1971 by K.J. Klepp. Cloning was slow and tedious, and even in the early days of PCR, the heat cycles would denature and polymerase requiring it to be added anew every cycle. PCR may be the single most indispensable technique used in modern biology. 1992 Genes Controlling Autography. Autography is a process of self-destruction and recycling in the cell in response to stress. Yoshinori Osumi has developed a method to study this process in yeast and has managed to find several genes responsible for this process. His work has contributed both to science and medicine as autography was found to play a significant role in neurodegenerative diseases such as Alzheimer's and Parkinson's. Osumi was awarded a Nobel Prize in 2016 for his work. 2007 CAS9 CRISPR system in bacteria. The team of scientists led by R. Barangu has discovered that bacteria use special enzymes to cut out pieces of infecting virus that are lately inserted into the bacterial genome. This system helps bacteria recognize viruses in time. Later, the scientists realized that those bacterial enzymes can be used for editing the genomes of other organisms. This discovery was crucial for cancer research and other fields. First, gene editing in humans was also performed using this technique.
So that's all for my report. Thank you for participating and listening.
Bawat bata may tanong,
Ba't ganito? Ba't ganon?
hayaang buksan ang isipan.